Hello everybody. This presentation is about biological hazards in food. My name is Matti Raasto and I work at the Estonian University of Life Sciences as a professor of food hygiene and veterinary public health. I have been working in this field for more than 20 years and I teach veterinary and food technology students in food production hygiene and food microbiological safety. Foodborne bacterial pathogens are my main research interest, but I have also worked with colleagues on the chemical food hazards and with natural antimicrobial compounds which can be used as antimicrobials and antioxidants in food. Each year in Europe, more than 23 million people are falling ill while consuming unsafe food. World Health Organization estimates that norovirus is the most common cause of foodborne illness in the European region with close to 15 million cases each year, followed by Campylobacter, which is responsible for almost 5 million disease cases. Salmonella causes the majority of contaminated food consumption-related death cases in Europe. Other major causes of death are Campylobacter, norovirus, Listeria monocytogenes, and Echinococcus multilocularis. Biological hazards in food can be pathogenic prions, viruses, bacteria, molds. Also, protozoa and other pathogenic parasites in food can cause foodborne diseases. The problem is that the presence of pathogens in food cannot be usually recognized by the senses. We cannot detect pathogens in food through the eyes or by smelling or taste. Pathogens can be present in foods that seem completely hygienic and safe to us. When microorganisms contaminate food, which causes foodborne disease, then these microorganisms are referred as foodborne pathogens. Foodborne diseases are caused either by infectious agents or by toxins, mostly enterotoxins. Foodborne diseases caused by biological hazards are either foodborne infections or foodborne intoxications. Most common foodborne pathogens are pathogenic bacteria such as Salmonella, Campylobacter, pathogenic Escherichia coli, Listeria monocytogenes, and others. There are also foodborne illness-causing viruses like noroviruses, hepatitis A and D viruses, rotaviruses, and astroviruses. Also, food-related protozoa such as Toxoplasma gondii, Cryptosporidium, Giardia, and additionally mycotoxin-producing molds can cause foodborne diseases. Not always heat treatment can eliminate biological hazards in food. This is because many microorganisms can produce thermostable toxins into the food. For example, enterotoxins can be produced by pathogenic E. coli, Clostridium perfringens, Bacillus cereus, Staphylococcus aureus, Shigella dysenteria, Yersinia enterocolitica, and Vibrio cholera. Bacterial spores can be even bigger food safety problem. Spore can survive high temperatures, for example, boiling for up to 5 hours. Also, they can survive disinfection, dehydration, and other treatments. When the conditions become more favorable, for example, presence of nutrients, water, suitable temperature, then the organism germinates from the spore and continues the multiplication in food. Spore-forming bacteria are some well-known foodborne pathogens like Bacillus cereus, Clostridium perfringens, and Clostridium botulinum. The question may arise, how can the microbiological food safety be ensured? We can say that the key issues in ensuring food microbiological safety are the effectiveness of the self-control programs, including prerequisite programs and food safety management systems. There is need for adequate hazard analysis and control. Validation and verification activities should be appropriate, and generally, HACCP systems need to be effective. Also, knowledge on food intrinsic, extrinsic, and implicit factors which influence the microbial growth in food is very important. And also, personal knowledge and positive attitude towards food hygiene and safety is one of the key issues in ensuring food safety. The most important intrinsic factors which influence the growth of microorganisms in food are pH, water activity, redox potential, and antimicrobial ingredients in food. The most important extrinsic factors influencing microbial growth in food are temperature, packaging-related gas composition, and relative humidity. Mostly the inhibition of microorganism growth in foods is achieved while combining aforementioned intrinsic and extrinsic factors. To prevent or minimize the biological hazards in foods, the food handler should use raw materials and food ingredients of high quality and from reliable suppliers. Implement good hygiene and good manufacturing practices and prevent contamination at all levels of food processing and handling. Comply with the rules of personal hygiene, including proper hand washing. Avoid bare handed handling of ready-to-eat foods and prohibit the handling of food by workers with disease symptoms or with skin infections. Apply minimum of plus 74 Celsius core temperature in food to destroy vegetative cells of pathogenic microorganisms present in food. Ensure the adherence to time and temperature regimes. Ensure the compliance with established food safety and process hygiene criteria. Verify that the self-control system is effective and food safety is ensured. This includes also food and environmental sampling to verify the compliance of food safety and process hygiene criteria. Some other activities may be required depending on production type and food safety risk level. Thank you for your attention. Please also check other study materials on the Safe Org Food project homepage.